My name is Joshua Mugabi. I welcome you to Topic 2, Nutrition in Plants. Our learning outcome B, assess the influence of environmental factors on the photosynthetic efficiency of plants to optimize photosynthetic rates and crop yields. We are looking at the learning activity B. In pairs, learners carry out scientific investigations on the behavior of gut cells under varying sucrose concentrations. Learners write a report including relevant drawings. Learners use their findings to explain the photosynthetic theory for the opening and closure of stomata. Behavior of gut cells and the photosynthetic theory. Item 1. We are going to focus on day-night observation of stomata. Students observe leaf epidermal peels of a bean plant at midday and at midnight. Stomata are open during the day but closed at night. Task. State the role of gut cells. Explain why stomata are open during the day and closed at night using the photosynthetic sucrose theory. State the effect of this behavior on photosynthesis. State the role of gut cells. Gut cells control the opening and closing of stomata. Explain why stomata are open during the day and closed at night using the photosynthetic theory, sucrose theory. During the day, photosynthesis occurs in the gut cell and surrounding mesophyll cells. Sucrose accumulates in the gut cells. Increased sucrose concentration lowers the water potential of the gut cells. Water enters by osmosis, making gut cells tagged. The stomatophores open. At night, photosynthesis stops. Sucrose is converted to starch or transported away. Water leaves gut cell. They become flaccid and stomata close. State the effect of this behavior on photosynthesis. Opening allows carbon dioxide entry for photosynthesis. Closing reduces water loss by transpiration. Still on behavior of gut cells and photosynthetic theory, item 2. Focusing on the effect of increasing sucrose concentration. In a laboratory experiment, epidermal strips are placed in a solution of different sucrose concentration. Task: predict the effect of high sucrose concentration inside gut cells. Explain this effect using osmosis. State the resulting condition of the stomatophore. Predict the effect of high sucrose concentration inside gut cells. Gut cells become tagged. Explain this effect using osmosis. High sucrose concentration lowers the water potential of gut cells. Water moves into gut cells by osmosis from surrounding cells. State the resulting condition of the stomatophores. The stomatophore opens. We are still on the behaviors of gut cells and photosynthetic theory, item 3. Focusing on the effect of reduced photosynthesis. A plant is kept in darkness for several hours. On observation, most stomata are closed. Task: explain the change in sucrose concentration in gut cells. Describe the movement of water in gut cells. Explain why stomata close under these conditions. Explain the changes in sucrose concentration in gut cells. Photosynthesis stops. Sucrose is converted into starch or translocated away. Sucrose concentration decreases. Describe the movement of water in the gut cells. Water potential increases. Water moves out of gut cell by osmosis. Explain why stomata close under these conditions. Gut cells become flaccid. Stomatophore closes, reducing water loss. Still under behavior of gut cells and photosynthetic theory, item 4. We are going to emphasize on relationship between light, photosynthesis, and stomato movement. Students compare stomato behavior in plants exposed to light and plants kept in shade. Task: explain how light indirectly affects stomata opening. Relate sucrose concentration to stomato movement. State one advantage of this mechanism to the plant. Explain how light indirectly affects stomata opening. Light increases photosynthesis. Photosynthesis increases sucrose production in gut cells. Relate sucrose concentration to stomatal movement. Increased sucrose lowers water potential. Water enters gut cells, making them tagged and opening stomata. State one advantage of this mechanism to the plant. Efficient gas exchange for photosynthesis. Reduced water loss during low photosynthetic activity. Still under behavior of gut cells and photosynthetic theory. Item 4. Focusing on application to drought conditions. During drought, plants tend to keep stomatas closed, even during day. Task. Explain how water stress affects gut cell behavior. Explain the interaction between sucrose concentration and water availability. State the adaptive significance of stomatal closure. Explain how water stress affects gut cell behavior. Water stress lowers water availability. Gut cells lose turgidity even if sucrose is present. Explain the interaction between sucrose concentration and water availability. Lack of water prevents osmotic entry into gut cells. Stomata remain closed. State the adaptive significance of stomatal closure. Reduces water loss by transpiration. Prevents wilting and dehydration. Marking emphasis. Collectively explain the photosynthetic sucrose theory. Link sucrose concentration to water potential and osmosis. Describe tagged and flaccid state of gut cells. Apply theory to light, darkness, and drought conditions. Use correct biological terminology. Conclusion. According to the photosynthetic sucrose theory, stomata opening and closure depends on change in sucrose concentration in gut cells. High sucrose concentration during active photosynthesis lowers water potential, causing water to enter the gut cell by osmosis and open stomata, while reduced sucrose concentration leads to stomatal closure. Thank you for participating in Topic 2, Nutrition in Plants. Our learning outcome B, assess the influence of environmental factors on the photosynthetic efficiency of plants to optimize photosynthetic rates and crop yields. Our learning activity has been B. In pair, learners carry out scientific investigations on the behavior of gut cells under varying sucrose concentrations. Learners write a report including relevant drawings. Learners use their finding to explain the photosynthetic theory for the opening and closure of stomata. If this has been useful, visit me at Mugabi Joshua Biology on YouTube.